3D correlative light and electron microscopy, or CLEM, would combine fluorescence with EM. And at Tigerberg, we have a large TB research unit. Tuberculosis is still a big disease burden in South Africa, and there's a lot of resources and research going into better treatment strategies and how to further understand this bacteria and this disease. So what happens during TB infection is at certain parts of the lung, macrophages or immune cells will start to aggregate. And this is what we see in this chunk here. So this is a slice of lung tissue that was fibrotome sectioned. And when we zoom into this area here, we took a stack on a confocal microscope. The TB was tagged with a red fluorophore. So the genetic variant of the bacteria that was expressing a certain fluorescent protein. We could take a 3D stack through that, overlay that with our eventual embedded part of the same region, zoom into the area that we knew definitively had TB, render out the cells that were infected, and here we have a multinucleated macrophage that contains TB. Now, importantly, we start to notice that the bacteria seems to be jumping from cell to cell during infection. So this is still an ongoing study that we're finishing up. And here I can give you a good example of the different kinds of scales. So initially, what happens here is mapping out of the whole granuloma, or the whole region of interest, essentially, that was acquired. And then you can zoom into that little part of overlay that we were confident in, that we knew that TB was situated in. All these little black globules are actually fat globules that pile up in the macrophages themselves. So we knew we were close. You can render out the individual cells. So this segmentation was done in elastic again, and rendering in a mirror. And then the individual TB I had to hand segment out, and the nuclei were pretty straightforward to segment. But now what you can do is you can compare cell by cell and map out the different nuclei, the cell borders, how much TB is in each cell. So you can think about this from a quantitative perspective. If you can map the density of bacteria per macrophage, you're getting to quite a powerful data set.